These are your video notes on viruses, and we'll start by talking about what a virus is. So a virus is a non-living particle. A particle, it's not even a cell. It's a non-living particle, um, and its really sole function is to just make copies of itself. They can't be seen with the kind of light microscopes that we might have in the classroom. You would need something called an electron scanning microscope to see a virus because they are so small. And those are very expensive, and classrooms are not going to have those. So we have to look at published and posted pictures that other people have taken to see viruses. To reproduce, they need to infect cells. They don't have their own machinery to make copies of themselves. They have the DNA or the RNA, but they don't have the ribosomes, for example, to make viral protein, right? They have to infect cells, hijack their ribosomes and their machinery to do it for them, right? And furthermore, right, they're non-living because of that reason. They can't reproduce on their own, but they themselves, again, they're not made of cells. They don't use energy. They don't have metabolism. They don't consume ATP, and they don't reproduce on their own. So they are considered non-living. However, they do evolve. Their DNA does mutate, and they are subject to forces like natural selection. So what are they made of? All viruses have genetic material. It is either DNA or RNA. Most of the ones you're familiar with are DNA viruses, but some, like HIV, are RNA viruses. And actually, the coronavirus, the one that we've just experienced and are currently still experiencing, is an RNA virus. The protein coat that surrounds that DNA or RNA is largely protective in nature. That's called a capsid. And usually around that, you have a lipid envelope that's basically derived from the host cell membrane. The virus doesn't make that on its own. It gets it when it leaves a host cell. But on the lipid envelope and on the capsid, depending on the type of virus, you've got these little proteins that serve to recognize and bind to proteins on the host cell. And this is how viruses, quote unquote, know where they are, because their surface proteins match surface proteins in your cells. And the whole reason that they do that is because actually the proteins and the membrane around the virus are in part derived from that host tissue. But for example, the influenza virus, which is what's shown over here, has little proteins that bind to surface receptors on your intestinal cells, and is one of the reasons why you get things like diarrhea when you have the flu. So 3 common viruses. The first only infects, I guess I was going to say affects, but infects and affects. It only infects bacteria. So this is called a bacteriophage. So the bacteriophage is the virus. The bacteria is what it infects. So despite how scary this lunar landing thing might look, it will never infect human cells. Its receptors do not match anything on human cells. It's just for bacteria. But you can identify all the parts here, I hope, if you want to pause and try and do that. Click through them right now for you. Right, here's the capsid, the head, the DNA or the genetic materials in the middle. This part is unique to this type of virus. This is the tail and the tail fibers, and this is where it attaches and injects its DNA into its host bacteria. Here's a tobacco mosaic virus, but again, the main parts are still there. You have the genetic material surrounded by a capsid, and the same thing here with the flu virus, right? Genetic material surrounded by a capsid, and there are surface proteins that serve to recognize and bind to surface proteins on the host cell. Here's a picture, electron scanning microscope picture that's been color coded. to show you the size, the relative size, and what this thing looks like. So it actually does look like a lunar lander. Here's your bacteriophage here. You've got the tail fibers attaching to the cell membrane, which is what's in green. The inside, the cytoplasm and its ribosomes are in light blue and all these other little dark blue specks and other portions. This is not a nucleus, by the way, and you should know it's not a nucleus because this is a prokaryote and prokaryotes don't have nuclei. So it's something else in the cell. I'm not quite sure what it is, but you can see the entirety of the virus, right? And you can only see a portion of the bacteria. So you can get a sense as to how small viruses are. And again, because they're not living things, they don't get a name. They just get some kind of number. So this is called bacteriophage T4, right? All right, so what's the life cycle of a virus? There's two components of the life cycle. The first is the lytic, and it is the one that you associate with being sick, right? So when you're sick and you have a you have a cough, right? The virus that has infected you is undergoing its lytic cycle. What are the steps? The steps are attachment. Now, I'm going to show you a picture here that involves bacteriophages and bacteria, but it's the same basic process for like the flu virus and human cells. So the virus attaches. Again, it attaches because it has matching proteins. I can add that on here. I'll say matching receptor proteins. And it's not like this thing is a heat-seeking missile. It's not searching or seeking out the surface of a bacteria that matches it. These things are bouncing around. They're diffusing around in the air. The key point here is they're diffusing. This is the random motion of molecules and water currents or wind currents or whatever media these things are passed through. And they just so happen to bounce into cells along the way. If they bounce into a cell and the receptors don't match, guess what? They bounce right back off. And this is why bacteriophages don't infect human cells, because their receptors don't match. But if they do happen to match, then they do bind. And that's really all it is. It's random motion and the interacting of these surface receptor proteins. If they match, then there's binding. And if they don't match, then the cell and the bacteriophage or the virus bounce off each other and they go about their merry ways. So assuming that they do attach, the virus attaches to the cell's membrane and injects its genetic material, right? That could be a DNA or an RNA strand. And from the host cell's perspective, it doesn't really know that it's viral DNA or RNA. So it is going to transcribe and translate it just like it transcribes and translates its own DNA. And that's basically why we say viral DNA or viruses hijack the host cell's machinery, right? They take advantage of the fact that ribosomes in the host cell are just going to transcribe and translate whatever DNA and RNA they find. Except that once a virus has injected its DNA or RNA into a cell, that cell will now be making viral proteins, right, in addition to its own proteins. So viral replication and assembly will occur. We can talk more about this if I see you again in AP Bio or whenever you have AP Bio or perhaps even Bio in college if you want to, about how this happens in more detail, but the gist is that as these proteins accumulate in the cytoplasm, their positive and negative charges on the amino acids, the polarity or the non-polarity of the different parts of the protein actually will spontaneously self-assemble into the viral components and the virus particles themselves. And then once you have the assembly of the viruses, they literally accumulate inside the cell and cause the cell to rupture or to lyse, L-Y-S-E. And lyse just means to burst or to break, which releases the viruses, yeah, but it also kills the cell in the process. So when you're sick, when you're coughing, when you're sneezing, when you have a fever, you have, assuming it's a viral infection, you have cells in your body producing viruses and they're exploding. And those cells are releasing other viruses into your bloodstream, into your lymph system, in your saliva, in your respiratory droplets, etc. Right? So this is when you would say, I am sick. right? I have an infection or an active disease. The lysogenic cycle or version of this viral lifespan, if you will, is nicer, I think is is a better way to say it. But a lot of the steps are the same. So the virus fuses to the host cell, injects its DNA, just like with the lytic cycle, there's nothing different yet. However, once the DNA is in the host cell, and we're using the bacteriophage example here still, notice that the DNA, the viral DNA, this pink portion, doesn't get transcribed or translated. It gets incorporated into the host genome. It becomes part of the host genome. So again, the key thing here is that it does not get transcribed. There's no transcription. And there's no subsequent translation into proteins. Those two things don't happen. So you're not going to get assembly of viral particles. What you will get, however, is every time this cell divides and undergoes mitosis or binary fission in the case of a bacterial cell, it copies the viral DNA or RNA as well, and the new daughter cells also have the new viral DNA or RNA in them. So I say it's nicer because you're not killing cells. This is not associated with an active infection. However, the virus still is passing on its genes, its DNA, because it's still getting copied. The DNA is still getting copied when the host cell copies. So no transcription or translation of the DNA, but the DNA is propagated into future generations whenever the host cell divides. And that's the lysogenic infection. The key thing here is that any virus can swap between these two infections, sometimes more predictably than others. Some factors like stress or dehydration or malnourishment contribute. A lot of times it's just harder to predict when viruses switch infection cycles. But let's just say, like for the herpes example, right? The type of virus that causes chickenpox, the type of virus that causes cold sores on your mouth, right? If you have a cold sore on your mouth, that little red spot along your lips, that's the virus undergoing the lytic infection. Those cells on your mouth, I'm going to just draw a pair of lips. That's a pair of lips. Just go with me here. And you've got a red spot, right? Those skin cells are popping, they're bursting, they're exploding, and there's little virus particles in all of that. Like, it's not water, it's mostly water, but all of the cytoplasm of your cells is rupturing, and that's really what all that kind of moisture and wetness is coming out of a spot like that. Sorry to gross you out. I hope you're not eating anything. But that's the lytic infection, right? But the cold sore doesn't last forever. Eventually, that red spot will dissipate, disappear, and it's not like the virus is gone. What has the virus done? Well, the virus has returned to the lysogenic cycle. It is actually dormant in the nerve cells. I'm going to try to draw someone's face now. It's dormant in the nerve cells actually at the base of the jaw. And that's where it kind of lies dormant in its lysogenic portion of its life cycle. And then perhaps when you're stressed later on, it reverts back to its lytic cycle. And it can do that for the entire course of your life. And viruses do this all the time. HIV can do this, where you get the HIV, you get the virus and you can have it for like a decade and not know about it because it's lysogenic. And then at some point, 5, 6, 7, 8, 9, 10 years later, it goes into its lytic cycle, and then you have AIDS, right? Then you have an active outbreak of the HIV or HIV virus. So know this diagram. I wouldn't necessarily memorize any of it per se, but just kind of know the basic steps and that viruses can and do swap back and forth all the time. Types of viruses, again, most are going to be DNA viruses. Let's see. Here's the herpes virus, chickenpox, and cold sores, etc. Smallpox was caused by DNA. These are all DNA viruses, but I do want to point out one specific type of virus called a retrovirus. And that's because retroviruses have RNA. They have RNA, right? And they're called retroviruses. The HIV virus, or HIV that causes AIDS is an example, and sometimes strains of cancer are actually caused by retroviruses. But if you look in the anatomy of a retrovirus, we'll look at HIV here. You don't have to know all of this by any means, but you should recognize, it has a viral envelope. I'm over here on the left, by the way, where the star is. It has RNA, as you would expect with a reverse or a retrovirus, but it has this reverse transcriptase. And this is the key thing here for a retrovirus, and this is the part that I want you to take away from this. Typically, I mean, not typically, like usually, the vast majority of the time, a cell is going to take DNA in the nucleus, it's going to transcribe that to messenger RNA, right? And that process is called transcription. I'm just going to abbreviate transcription. And then it'll go from messenger RNA to a protein, right? And this is called translation. You guys should know this by now, right? It is possible to go backwards from messenger RNA, or from an RNA molecule, to go backwards into DNA, then to go forward again into messenger RNA. And that's actually what happens with retroviruses. Because they have RNA, they inject RNA into the host cell. The host cell then reverse transcribes the viral RNA into what's called cDNA. You don't have to know this, but complementary DNA using an enzyme called reverse transcriptase. It's a very, very easy name to remember because it tells you exactly what it is. It's doing reverse transcription, right? It's going backwards from RNA to DNA, and then ACE is telling you it's the enzyme, right? So it's a nice, easy name. But this is a process unique to retroviruses, and retroviruses are the only viruses that they actually contain their own copies of reverse transcriptase enzyme, because regular host cells don't use reverse transcriptase. It's unique to these types of viruses. So just be aware of it. These are also the types of viruses that incorporate their genome into yours permanently. Other viruses that are DNA based, that genome does not incorporate into yours usually, but for retroviruses it does. How to defend against viruses? Let's wrap this video up. So the only defense against a virus, at least now that we have, is a vaccine. So a vaccine is a weakened solution that contains usually a portion of the virus or an inactivated virus. And we can inactivate the virus because we can cause damage essentially to the proteins that it needs to replicate, or to the proteins that are on its surface and it can't bind correctly with the host cell. They are not active infectious viral particles. You are not going to get the actual illness or disease or virus from a vaccine. Furthermore, and it goes without saying, that there is no relationship between getting vaccinated or having autism or anything like that, which we can talk more about in class. Vaccines are incredibly effective at preventing the disease that they're associated with. To be in a public school, for example, you actually have plenty of vaccines, and there are probably diseases you've maybe only heard of in passing because vaccines have been so effective at getting rid of them, like measles, mumps, rubella, or polio. You don't have to worry about those anymore. You don't have to worry about chickenpox because you've had a chickenpox vaccine. So, and we were in the middle of a pandemic. We've had the coronavirus vaccine, and that's actually an mRNA. It's a new type of vaccine where we don't inject any part of the virus at all, not a damaged portion of it or a damaged entire virus. We actually don't even inject any viral protein at all. We've been injecting messenger RNA directly into your cells, which are then translated into the spike protein. If this is the surface of the cell or the surface of the virus, this spike protein that kind of looks like a club. I tried to draw it looking like a club. I'll draw it a little bit better. Is what is produced by your body, and we can talk more about this in class, when you have something like an mRNA vaccine for coronavirus. So have that. So basically you're training your body to recognize foreign invaders so that if ever in the future you see the virus or a portion of the virus or a component of it, like with a second dose of a vaccine, for example, your immune system's like, hey, I recognize that, and it's already got a whole bunch of antibodies ready to go to attack it. So again, the whole purpose of a vaccine is to develop antibodies in the event that you actually see the virus later on. And you have a much better chance of getting it rid of or getting it out of the body so that it doesn't even have the opportunity to infect cells. All right, why don't we have vaccines for all viruses? Well, the first is that the surface proteins on a virus change rapidly because they mutate. So vaccines are really only effective against proteins that don't change very often. If you have a surface protein that doesn't really mutate, then a vaccine can be effective for decades or centuries. If you have, and that's been true for things like smallpox, measles, and polio, you don't have to have repeated vaccines, you just have the one vaccine and you're done, right? However, with something like the cold, which mutates so quickly we have no vaccine for it, or the flu, which still mutates quick enough to where we have to have a new vaccine every year, those do mutate pretty quickly, and you do have to routinely take vaccines. And vaccines from one year might not protect you the next year, right? And that's why you have to get flu shots repeatedly, because again, the flu does mutate very, very quickly. Antivirals. You can take antivirals to treat viral infections, but you're not going to cure the infection necessarily. You can stop its progression for all intents and purposes, but you're not going to get rid of the viruses already present. And the reason for that is antivirals affect proteins that the virus needs to reproduce, right? For example, if you have HIV in your system and you are on antivirals, the antiviral medications are preventing HIV from replicating in part by blocking reverse transcriptase. Right? We've already talked about how HIV is a retrovirus. So if you take an antiviral for something like HIV, that antiviral blocks reverse transcriptase. It doesn't disassemble the viruses already present in your body. So that's what I mean when we're saying that they don't cure the infection, but they do kind of prevent the spread of it or reduce the proliferation of virus already in you, right? So antivirals attack the enzymes or proteins needed for replication, but not the actual virus itself. Actually, that's about it. So we can talk about this more in class as you guys want, and I will see you then, and I hope this helped.